The first question we discuss is digestion and absorption of carbohydrate. Dietary carbohydrate consists of polysaccharide, disaccharide and monosaccharide. This polysaccharide and disaccharide, they have to be hydrolyzed into monosaccharide because only monosaccharides are absorbed by the intestine. The process of digestion of carbohydrate, it begins from the mouth. Salivary gland secrete an enzyme, salivary alpha amylase, which will act on the alpha 1,4 glycosidic linkages. This enzyme cannot act on the alpha 1,6 linkages which is present on the branch points. This enzyme will convert polysaccharide into dextrins. Very little digestion occurs in the mouth because food remains in the mouth only for a shorter period. And the carbohydrate digestion, it is temporarily stopped in the stomach due to the high acidity. Only some amount of sucrose is converted into glucose and fructose by the action of HCl. The further digestion of carbohydrate occurs in the small intestine by the enzyme pancreatic alpha amylase. Pancreatic alpha amylase convert dextrins into maltose. The brush border of the intestinal cell contain different enzymes like maltase, sucrase and lactase. These enzymes will act on the corresponding disaccharide and convert it into monosaccharide. For example, maltase will convert maltose to 2 molecules of glucose. Then sucrose, it is converted to glucose and fructose by the enzyme sucrase. Then lactose, it is converted to galactose and glucose by the enzyme lactase. Then these monosaccharides are absorbed. The disorder associated with digestion of carbohydrate is lactose intolerance. Lactose intolerance, it is due to the deficiency of enzyme lactase. Since the enzyme lactase is deficient, lactose get accumulated in the gut. That will lead to irritant diarrhea and flatulence. Coming to the absorption of carbohydrate, only monosaccharides are absorbed in the intestine and the absorption rate is maximum for galactose, moderate for glucose and minimum for fructose. Glucose has specific transporters which are transmembrane protein, and the absorption of glucose, it involves different mechanism. Absorption from the intestinal lumen into the intestinal cell is by co-transport mechanism. A membrane bound carrier protein carries glucose along with sodium. So this is the intestinal lumen and this is the intestinal epithelial cell. The absorption of glucose from the intestinal lumen into the intestinal epithelial cell, it is with the help of a carrier known as sodium dependent glucose transporter 1. This sodium dependent glucose transporter 1, it has 2 binding sites, one for the sodium and the other for glucose. So you can see 2 binding sites. So this sodium dependent glucose transporter, it has 2 binding sites, one for the sodium and the other one for the glucose. Binding of sodium to this carrier protein, it will bring a conformational change. This conformational change enables the binding of glucose. Then the sodium, it diffuses into the cell, dragging glucose along with it, and energy is indirectly provided by ATP. The same intestinal epithelial cells, they have a different transport mechanism on the membrane facing to the capillaries. That is, these intestinal cells, they will release glucose into the bloodstream by a carrier mechanism called glucose transporter type 2. And this transporter, it is not dependent on sodium. It is a uniport facilitated diffusion. That is, glucose binds to the transporter on one side of the membrane. When fixed, the complex will change its configuration. This will lead to the closure of the first binding site, and at the same time the binding site is now exposed onto the inner side of the membrane, releasing glucose. So at this side, so you can see at this side you have a different glucose transporter, that is glucose transporter 2. Glucose transporter 2, it is not dependent on sodium. It is a uniport facilitated diffusion, that is glucose binds to this glucose transporter, that will lead to the closure of the first binding site. and at the same time the binding site is now exposed onto the inner side of the membrane, releasing glucose into the blood. So the intestinal epithelial cell at the intestinal lumen, the glucose transport is by a co-transport mechanism, that is glucose is absorbed along with sodium, and at the basal surface another transporter, that is GLUT2 is present. At the luminal side, glucose absorption is with the help of sodium dependent glucose transporter, and at the vascular side it is by glucose transporter 2. Glucose transporter 4, it is present in skeletal muscle and adipose tissue, and it is under the control of insulin. Insulin will induce the movement of intracellular glucose transporter 4 molecule to the cell surface and increases the glucose uptake. The other glucose transporters are glucose transporter 1, it is present in RBC, brain and kidney. Glucose transporter 2 is present on the serosal surface of intestinal cells, liver, beta cells of pancreas. And this is for the glucose uptake in liver, and it is a glucose sensor in beta cells. Glucose transporter 3 is present in neurons and brain. Glucose transporter 4, it is present in skeletal muscle, heart muscle and adipose tissue, and this is under the control of insulin. Glucose transporter 5, it is present in the small intestine, testis, sperm and kidney, and glucose transporter 5 is a fructose transporter and it has poor ability to transport glucose. And sodium dependent glucose transporters are present in intestine and kidney. In intestine, sodium dependent glucose transporter 1, and in kidney, sodium dependent glucose transporter 2 is present. Sodium dependent glucose transporter 1 is for the absorption of glucose from intestinal lumen into the cell. Glucose and galactose, they are absorbed by same transporter, that is sodium dependent glucose transporter. As we said, it is an energy dependent process against the concentration gradient, and therefore absorption is almost complete from the intestine itself. For fructose, it is absorbed by glucose transporter 5, and other monosaccharides, they are absorbed by carrier mediated facilitated transport. Classify carbohydrate with examples. So carbohydrate, they are polyhydroxyaldehyde or ketone or compounds which yield these on hydrolysis. And they are classified into monosaccharide, disaccharide, oligosaccharide and polysaccharide. So monosaccharide, they are the basic building blocks of all carbohydrate, and they cannot be further hydrolyzed into simple compounds. And based on the number of carbon atom, this monosaccharide, they can again be classified into trioses, tetroses, pentoses, hexoses, etc. And based on the functional group they are classified into aldoses and ketoses. Aldoses are monosaccharide which have aldehyde group, example glucose, and ketoses are monosaccharides having a keto group, example fructose. And when two monosaccharide units are combined together by a glycosidic bond, that is known as disaccharides, and these disaccharides include sucrose, lactose, maltose, and here sucrose is a non-reducing sugar, and the sucrose, it is made up of glucose and fructose. Lactose, it has one molecule of galactose and one molecule of glucose, and it is connected together by beta 1,4 glycosidic linkages. And maltose, it contains two glucose residues, and the linkage present is alpha 1,4 linkage. And when there is 3 to 10 monosaccharide units are combined together by glycosidic linkages, that is known as oligosaccharides. And polysaccharide means when more than 10 or more sugar molecules are attached, then that is known as polysaccharide. Example for polysaccharides are starch, glycogen, cellulose, etc. So monosaccharide, they are the simplest form. They cannot be further hydrolyzed. They can be again classified depending on the number of carbon atoms present. If 3 carbon atoms are present, it is known as trioses, 5 means pentoses, like that. And based on the functional group, it can be classified into aldoses or ketoses. If aldehyde group is present, that is aldoses, and if a ketone group is present, that is ketoses. And disaccharide, that is 2 monosaccharide units are combined. Disaccharide include maltose, lactose and sucrose. You have to study which are the monosaccharide units present and what is the linkage. Example for trisaccharide is raffinose and tetrasaccharide is stachyose. Polysaccharides, it is classified into homopolysaccharide. Homopolysaccharide means same monosaccharide. It is composed of single kind of monosaccharide. Example, starch, glycogen and cellulose. Heteroglycans means they are composed of 2 or more different types of monosaccharide. Example, hyaluronic acid, heparin, etc. Define mucopolysaccharide and give 2 examples. Mucopolysaccharide or glycosaminoglycans, they are heteropolysaccharide containing uronic acid and amino sugars. Acetylated amino group, sulfate and carboxyl groups are also present. Because of the presence of these charged groups, they attract water molecule and produce viscous solution. Second part of the question is give 2 examples. You can write any 2 examples like hyaluronic acid, heparin, chondroitin sulfate, dermatan sulfate and keratin sulfate. So hyaluronic acid, it is present in connective tissue, tendon, vitreous humor, etc. It serves as a lubricant in joint cavities and it is composed of repeating units of N-acetyl glucosamine and glucuronic acid. Then chondroitin sulfate, it is present in the ground substance of connective tissue which is widely distributed in the cartilage, bone, tendon, cornea and skin. It is composed of repeating units of glucuronic acid and N-acetyl galactosamine. Then dermatan sulfate, it contains hyaluronic acid and N-acetyl galactosamine. It is found in skin, blood vessel and heart valve. Then heparin, it is an anticoagulant. It contains repeating units of sulfated glucosamine and alpha-1,4 hyaluronic acid. Then keratin sulfate, it is the only glycosaminoglycan which does not contain any uronic acid. Here the repeating units are galactose and N-acetyl glucosamine. It is found in cornea and tendon. Next question is name the enzyme deficient in lactose intolerance. The enzyme deficient in lactose intolerance is lactase.